A distinguishing feature of humans is the volume of our brain relative to our ancestors and to other non-human primate species. So let's consider what's known about how the evolution of human brain size evolved. To start, I'm going to give you background from this paper, which provided this figure, which shows how far in the past, as usual, this is a phylogenetic tree with time running to present at the bottom in this case. So we've got the common ancestor of all of those different lineages at the top. And we see that among all of these lineages of primates, humans have a much larger brain volume in terms of cubic centimeters than all of these other species. So it's likely that something happened, specifically in the human lineage, since our common ancestor between humans and chimpanzees, that prompted or caused this increased brain volume. So now let's look at how positive selection has acted on the human genome. First, I want to show you a couple of slides where this paper listed most or all of the human genes that have undergone positive selection since our common ancestor with chimpanzees. And remember, positive selection means these are genes in which mutations are favorable, that diversity in the genetic sequence, and then probably also the amino acid sequence of the proteins, is beneficial. So let's take a specific look at a few examples. First, ApoBec3G. This is a protein that's an antiviral protein, and it specifically helps us, well, individuals that have particular variations of ApoBec3G resist or prevent HIV infection. So that is an immune-related gene. So most of these host pathogen interaction genes kind of make sense in terms of the benefit of positive selection. We would expect immune-related genes to favor variation. We know that our major histocompatibility loci, or MHC genes, we benefit from them having variation. Our immune system, our antibodies, benefit from being diverse. Now we go on to this reproduction category. So what can we find in here? Well, interesting enough, we have some zona pellucida proteins, and those are proteins that are expressed on the oocyte, and those are proteins that interact with sperm to ensure that the sperm that's contacting the egg is from the same species to prevent interspecies fertilization. So again, these are reproductive related genes and proteins. It kind of makes sense for them to undergo positive selection. They differ vastly between species. So these types of genes are expected and are observed to undergo positive selection, diversifying selection. We have things like melanocortin-1 receptor, which is responsible for pigmentation in skin, which we know varies a lot between different human populations. So that's perhaps not surprising that it undergoes positive selection. And we also have things like sensory systems, olfactory receptors and opsins. Olfactory receptors are proteins that are expressed in our noses that help us smell different smells. The opsins help us observe different wavelengths of light. And the VNO, or vomeronasal organ receptors, sense pheromones. So it's interesting to note that in primates, those pheromone detection proteins have evolved rapidly. Finally, for now, related to today's video, two brain anatomy related genes, ASPM and MCPH1, have undergone positive selection, specifically in the human lineage, and those are what we're going to look at today in terms of the evolution of human brain size. So we're first going to look at how these genes are identified as being involved in brain development, and that's going to use the approach of genetic mapping. Then we're going to look at another molecular evolution technique that helps us determine whether or not mutations were beneficial or neutral. So to start, let's look at how microcephalin, the gene MCPH1 specifically, was identified in this paper. So the next few slides are going to come from images from the paper, aside from this one, which came from Online Mendelian Inheritance in Man. And so these I'm just showing you to give you an idea of what the trait of microcephaly looks like. It's reduced brain volume in affected individuals. In particular, it's a reduced cerebral cortex volume, and here we can see some ways that the cerebral cortex functions in humans. So the way the authors originally identified this gene was they first started with two different families, two unaffected parents, the square is the father and the circle is the mother, who had an affected daughter and an affected son. These were individuals with microcephaly. So this immediately looks like a recessive disorder because it's not present in the parents, but it shows up in their offspring, and we know recessive diseases tend to skip a generation. So what the authors do for genetic mapping is you take usually unrelated individuals, like these two families, they're not related to each other at all, and you look in the affected individuals, which is described in this black box, for regions of a particular chromosome, an unknown chromosome. You look through all the chromosomes, look for regions where the affected individuals share the same genotypes, which is what's shown in the black box in this original paper. So we can see that that haplotype is present in one copy in the father, but not both, and also in this mother. And so that's how this individual, through the egg and the sperm of the parents, inherited homozygosity for this haplotype. So let's step back for a second. Let's take one of these rows for a second, like D8S1798. That is the nomenclature for a specific spot on, in this case, chromosome 8 in humans. And these numbers, 1 and 1, are alleles of that locus. So this is most likely a microsatellite, which is where a particular DNA sequence, like let's say AT, repeats multiple times. So if there was an AT2, that would suggest that at this spot on this chromosome there are 2 repeats of the AT unit or monomer. If there was an AT3 allele, that would be AT. So if there are different numbers in an individual, these are diploids, these are humans, each human has 2 copies of each chromosome. If there's 1 and 1, that suggests that this individual is homozygous for whatever number of repeats at this locus, D8S1798, this spot. So in other words, what these vertical representations are the linear representation of the DNA sequence of human chromosome 8, and the 2 copies of chromosome 8 that, in this case, this daughter has. So, to step back to the original interpretation, this daughter is heterozygous here on chromosome 8. She's a 1/5 or a 5/1. Heterozygous, homozygous 1, homozygous 3, homozygous 2, homozygous 4. And then she's homozygous for all of these different loci, regions of chromosome 8. And this other unrelated individual is also homozygous for all of those loci. But outside of this black box, the 2 individuals differ, homozygous for allele 1 versus homozygous for allele 4. And so it's the fact that these individuals are not directly related to each other, but they share the same trait, microcephaly, and they share the same genotype at this region on this chromosome, this tiny region, about 2 million base pairs of DNA in this black box, represented on chromosome 8. And presumably, although the authors don't specifically mention this, this is the only one of the 23 chromosomes 1 through 22, and then the X chromosome, where these two individuals actually shared the same genotype. So what that suggests in genetic mapping is when there is the same trait and the same genotype, that probably suggests that there's a gene somewhere in this 2.1 million base pairs of DNA that has a specific mutation that accompanies this set of alleles of these other loci that causes the trait. Now, the authors in this case got lucky in finding that there was only 2.1 million base pairs in this region, and this region only contains 2 known genes. One is angiopoietin 2, which is a gene that is a growth factor for angiogenesis, for blood vessel and artery growth, which may or may not be involved in expansive brain volume. Certainly if you have more brain volume, you would need more blood vessels and capillaries and arteries. But there was also the second gene, a novel gene of unknown function. That is, there's a coding region in this region, but nobody knew what it did. So when the authors looked at that gene, which winds up later being MCPH1, microcephalin 1 is the name, they call this unknown gene. It has a point mutation in the second exon that causes a serine at the 25th amino acid to become a stop codon, which means that the normal protein has that sort of structure, but the mutant protein, anybody who inherits this specific point mutation, a single nucleotide change, makes a truncated version of the protein MCPH1. And that is the mutation when homozygous, when you have 2 chromosome 8, and both of your alleles have this mutation, that causes microcephaly. And so they looked at this in panel C by looking at DNA sequences of this specific region, and they're just pointing out that the control individual has a TCA at this specific position. That's a serine codon, where that C to T mutation causes a TGA, which is a stop codon. So this point mutation in the coding region of this gene is stronger than likely causes microcephaly, or microcephaly, rather. So next, the authors wanted to look at a few other pieces of information to really ensure that this is the causal gene, MCPH1. So they wanted to know where in the organism this gene is transcribed, because presumably if MCPH1, this novel gene that they've just identified, is causing microcephaly, it better be expressed in the brain and not in other tissues. Well, in this case, they looked at transcript levels in various tissues, and they find that it is expressed in the brain, but also in a few other tissues. And that's not a problem. At least it's expressed in the brain, so that further suggests that this gene, MCPH1, could actually be causing this change in brain development. And down here on the bottom we see the positive control. So GAPDH is expressed in every tissue. They also want to make sure that it really is expressed in the brain. They want to know where in the brain this gene is expressed. So in this case, this is an in situ hybridization experiment, as we've seen before, where they're looking at transcript levels in slices of segments of brain. So they can see that there's expression of this gene around the ventricle in this case. So this gene is expressed in the brain, and this is mouse brain, by the way. And so it, again, makes it very likely that this gene could be involved in producing microcephaly. Now let's review molecular evolution analyses that we've looked at previously. We've looked at how molecular evolutionary biologists or geneticists detect the act of selection, positive or diversifying selection versus negative or purifying selection, on new mutations that occur in genes. And we've seen this ratio of dn, the number of non-synonymous changes per non-synonymous site, over ds. And we've looked at the idea that if there are more non-synonymous changes than synonymous, that suggests that those non-synonymous mutations are beneficial, and that suggests, again, positive or diversifying selection. And we just looked at a few examples of genes in humans that undergo positive selection. If the two ratios, or if the two values are the same, which makes the ratio 1, that suggests that no form of selection is currently, or was in the recent past, acting on the gene. And if the ratio is less than 1, that means there are more synonymous mutations per synonymous site than non-synonymous or amino acid changing mutations. That suggests that while mutations occur, the ones that are allowed to exist and pass from generation to generation are more frequently synonymous changes, the ones that don't change amino acid sequence. And that suggests that that gene is undergoing or experiencing purifying selection. The changes to that gene are bad, and when they happen, they're being gotten rid of or weeded out through natural selection. So I'm not going to talk about this slide because we've already seen this slide, but I want to point out that there's a drawback to using just DNDS analysis, and that is because that is usually used to compare individuals within a species. But when you use DNDS to compare positive or negative selection between species, there's this issue that differences could be caused by random mutations, or it could be caused by a form of selection, either negative or positive. So a new test, not the DNDS test, but the McDonald-Kreitman test, or the MK test, can help distinguish whether or not changes between species are caused by neutral processes. So here's a quote from the original McDonald and Kreitman paper. They say that if protein evolution occurs by neutral processes, that means mutations are random and don't positively or negatively affect the protein, then we'd expect the ratio of replacement to synonymous fixed substitutions to be the same as the ratio of replacement to non-synonymous polymorphisms. So I want to stop and clarify this terminology. This is going to look very much like the DNDS test, but here a substitution is a fixed change between species. And what I mean by fixed is that that difference is identical. So all of one, so let's say we've got species A and species B. And if we look at one spot, the same spot on one chromosome, all of the individuals in species A have, let's say, a G there at the second nucleotide. And if we look at the same spot in species B, all of the individuals of that species have an A. So that is what's called a fixed change, because the difference is fixed, it's complete, where everybody, it's diagnostic. Everybody of species A has a G. Everybody in species B has an A. That is what's called a substitution. Then there are also within species changes. So differences of individuals within species are called polymorphisms. So that would be if we take, let's say we have two individuals A1 and A1 from species A, and two different individuals from species B. So if we start with the same DNA sequence we had above, everybody in species A has that G, everybody in species B has an A at the second position. And if we make these go a little bit farther, we can can see that these are the same DNA sequences, but there might be a mutation there, and likewise there might be a mutation there. So what I'm focusing on here is, although it's typical for any individual in either species to have a G, there's sometimes a within species polymorphism, where there's been a unique mutation in one individual in that population or in that species. So those are polymorphisms. So what the MK test is going to look for is whether or not there are changes in the number of fixed changes between species, and differences within species. And the null hypothesis, that is, if there's no selection acting, that the ratio of non-synonymous to synonymous D, which is what we've looked at before, should be equal to the ratio of polymorphisms. So here's another example of that. So let's focus down here in the bottom left first. Each asterisk represents a fixed difference, and each cross, or dagger, represents a polymorphism. So in this case, from a common ancestor, there are two different species. There's one lineage of species B, and then there are different populations of species A. And in this case, the asterisks represent those fixed differences between the two species. So you can think of each of these asterisks as a new mutation that causes a DNA sequence difference between the species. Likewise, the daggers or crosses are four different unique mutations that occurred in different lineages of populations of the same species. So the fact that there are an equal number of asterisks or differences between species and daggers, differences within the species, suggests that whatever gene this is we're looking at the sequence of is not experiencing any form of selection, that mutations can fix that distinguish the two species just as frequently as mutations occur within the species. So that is a neutral type pattern in the MK test. Now we have the two different versions of this, where there's a single fixed difference that distinguishes the two species. But within species A, there's a lot of genetic divergence. There's a lot of diversity within the species, but there's not very much genetic difference between the two species. And what that suggests is that there's purifying selection. And the way that is suggested is that there are lots of mutations that occur in this gene, whatever gene it is, but they don't last long enough in evolutionary time to distinguish species A from species B. And that's why there's a low number of fixed differences. So if a gene is experiencing purifying selection, which is the case in this example, we expect a lot of mutations to occur at random, but we expect that most of those to be gotten rid of by natural selection, so that you get lots of variation, but very little of it is maintained over longer evolutionary times. Likewise, this other example of positive selection, or diversifying selection, also sort of seems counterintuitive, I think, is that what we see in this case is the opposite pattern, where there are lots of fixed differences between species. So these two species look very genetically diverse, but when you look within species A, you don't see that much genetic diversity. There's maybe 1 point mutation there that distinguishes population 2 from all of the other populations. And what this signifies is that there's a rapid acceptance, so to speak, of mutations. That whenever a new mutation happens in one of these populations, that is highly favored, and so it spreads through the populations rapidly and fixes and becomes one of these asterisks that distinguishes all of species A from species B. So there's very little genetic diversity remaining in the current populations, because every time, or frequently anyway, most of the time, some of the time, when a new mutation occurs, that's beneficial, and so it becomes a fixed difference and doesn't remain as a polymorphism. So that's how the McDonald-Kreitman test analyzes these. So again, if the ratio of between species synonymous and non-synonymous mutations, DNDS, is the same as the within species polymorphism, that suggests that there is no selection happening on this gene that we're studying. And then if the DNDS ratio is less than the PNPS, the within species ratio, that suggests negative selection, and the reciprocal pattern suggests positive selection.